And we're right on time to move into our last presentation for the CEDAR project. So next, I'd like to bring back Zeynep, our cancer bioinformatics instructor, to present the Neoepitope and prostate cancer meta-analyses. Thanks so much, Zeynep.
So now I would like to show you some of the analysis that we have performed using the data that we have in CEDAR actually. So when we first started building CEDAR, we wanted to test how useful it is and its utility on two very different types of cancer antigens. So we created, so to say, two prototype data sets. We focused first on prostate cancer antigens because these are shared, non-mutated epitopes. They're associated with prostate cancer, which is a cold tumor. So these are mostly classical tumor antigens, like overexpressed ones or tissue-specific ones. And then the second data set was the neoepitopes. These are mutation-derived epitopes, usually from highly mutated hot tumors, and they're often used in personalized cancer vaccines. So the data is very diverse. So these two subsets present orthogonal categories. One is rooted in decades of tumor immunology and very well-characterized antigens, and the other is centered around emerging patient-specific targets that are harder to validate, and you have less assays per epitope and antigen. So once we had enough data for both of these categories, we performed meta-analysis. So we wanted to illustrate the capabilities of CEDAR, and then also we wanted to use that information to refine our curation rules, our fields and parameters. So it was a really useful exercise to really dig into these two subsets of antigens. And then also the goal was to see if we can find any patterns, biases, and knowledge gaps in the existing cancer epitope literature. Okay, so let's start with the prostate antigens. So here, these are actual tables from the paper. I will show you the paper later as well. You can take a closer look. So here you see a breakdown of the data we had in CEDAR for prostate-associated epitopes. We have a total of 8 prostate-associated antigens, which are listed here. From these antigens, we have 278 epitopes, and they were curated from 130 publications. The 3 most studied antigens are PAP, PSA, and PSMA. They account together for more than 2/3 of all the prostate antigen epitopes. Interestingly, the total number of epitopes did not correlate with protein length. Instead, it mostly tracks with the number of publications available per antigen. So this is again related to the question of biases. So these are just proteins of interest, and they are just being studied in more publications. So we have more data for them. Next, we had a look at the T-cell assay types for epitopes from these top 3 antigens. So overall, they captured nearly 1,400 assays across the several assay classes, including the T-cell assays, tetramer staining, and MHC ligand assays. So the class 1 restricted epitopes dominate the data, especially for PSA and PSMA, but there's also surprisingly a number of class 2 restricted epitopes, particularly for PAP. So the tetramer data is very sparse. Only a handful of epitopes have been validated with tetramers, actually. Next, we look at the specific cytokines that were measured in the T-cell assays, and we can clearly see that interferon gamma dominates across all of the antigens, and PSA is the only antigen tested across a broader panel of cytokines. But these numbers are also only single digits, so it's a lot lower than the interferon gamma assays. So yeah, for the prostate antigen, a quick conclusion. The literature is very skewed towards PAP, PSA, and PSMA, leaving the other 5 antigens under-characterized. And T-cell assays are mainly restricted to MHC class 1 epitopes, So this is another gap for the MHC class 2 epitopes that might also be relevant. And then the cytokines other than interferon gamma are not well studied. We can take away from these that like additional studies would be very useful to really get a comprehensive characterization of prostate antigens, starting with the other undercharacterized antigens, looking at MHC class 2 epitopes and assays as well, and then also not only focusing on interferon gamma, but also the other relevant cytokines. So I'll now switch to the neoantigen meta-analysis with a brief introduction about neoantigens. So cancers, they actually accumulate mutations in their DNA so that they can acquire their cancerous capabilities and grow indefinitely. And if a mutation is in a coding region, it can get translated into a mutated protein, and then it will get chopped up into mutated peptides, and these peptides are then called neopeptides. So these neopeptides can get presented on MHC on the surface of cancer cells and then get recognized by T cells as foreign because they are mutated and different from self. And if they get recognized by T cells, we call them neoepitopes actually, because they can induce an immune response. So why are neoepitopes so important? On the clinical side, we have multiple studies now showing that the number of neoepitopes is associated with better outcomes in patients receiving checkpoint blockade therapy. And there are therapies directly targeting neoepitopes with vaccines, for example, and they have been showing great promise. We also have shown in a recent study that spontaneous T-cell responses against neoepitopes can be detected across a variety of different cancer types. But then we have the computational challenge predicting which mutations will give rise to neoepitopes that are actually recognized by T cells still remains a major challenge. And there has been a lot of work. There's many publications coming out trying to understand what makes a neoepitope actually immunogenic and how we can improve the predictions and develop better tools. And this is exactly where our meta-analysis comes in. So by systematically analyzing the experimentally validated neoepitopes that we have in CEDAR, we can better characterize their features and ultimately contribute to improving these predictions. So what makes our analysis so unique is that this is the largest meta-analysis of neoepitopes that have actually been experimentally validated, curated from the literature. So because previous studies often relied on predicted neoepitopes, which does not give the accuracy that we have now with all these experimentally validated epitopes. So a lot of different data is available in CEDAR, but for this meta-analysis, we focused specifically on human neoepitopes that result from single nucleotide variants in cancer patients. So we excluded animal models, we excluded cell lines, we specifically only included the assays that were tested in cancer patients. In total, we analyzed over 1,600 neopeptides from 180 published studies across 28 different cancer types. One important point is that a single peptide can appear in multiple studies. So for example, we have the peptide. It has been tested in 3 different T-cell assays. 2 have been negative and 1 has been positive. So to standardize our analysis, we considered a peptide to be a validated neoepitope if at least 1 assay showed T-cell recognition. For example, if we only had 1 T-cell assay that was positive, that was enough to say this is positive. And if we only had 1 negative assay, this was considered also negative. So following this classification, we found that about 13% of neopeptides were classified as neoepitopes, and the remaining 87% showed no T-cell response. So we began by looking at the top 100 proteins with the most tested neopeptides in CEDAR. Here, each column represents one of these top 100 proteins. On the top, we have the number of neopeptides and neoepitopes in red and green. And in the middle, we have each protein's length. And in the bottom, we see the mutational frequencies. How frequent are these genes actually mutated in cancer patients in a CBio portal? And we can see that many of these proteins, the top 100 ones, are either like very large, as we can see here, we have some spikes, or they come from recurrent mutations, as we can see by these spikes in the CBio portal data. We actually also found a significant correlation between protein length and the number of tested neopeptides, which makes sense because the larger the protein is, there's just more sequence that the mutation can occur and generate a neopeptide. However, when we look at the number of neoepitopes that had a T-cell response, this correlation is much weaker. So this shows while large proteins tend to produce more neopeptides overall, they are not necessarily more likely to be recognized. Instead, we found that driver genes are significantly enriched for neoepitopes, as we can see here in this plot. So these findings might suggest that the immune system may specifically recognize driver genes, perhaps of the central role they play in tumor biology, and because they are under selective pressure. Because we saw that TP53 has the most neoepitopes, we wanted to investigate them in detail. And actually, TP53 is the most frequently mutated gene in human cancers, across all the human cancers. So it's not surprising that we also have the most neoepitopes. In total, we found 152 unique neopeptides, and 35% of them were validated as neoepitopes. And so 35% is significantly higher than the global average of just 13% across all the proteins. But again, we have the bias here because TP53 is being tested again and again. So these epitopes have a lot more assays. So the chances are higher that you have a positive assay as well. So here in this plot, you can see the length of the TP53 protein. And then these bars represent the different neoepitopes and neopeptides across the TP53 sequence. So the red ones are the negative ones and the green ones are the positive ones. And yellow shows where the mutation is located within the peptide. And we can see that the majority of epitopes are located right here in this P53 binding domain. And this domain is actually the critical part of the protein that plays a role for the role as tumor suppressor that TP53 has. So it makes sense that the cancer mutates this domain more often, and if you have more mutations, you can generate more neopeptides and neoepitopes from there. And we specifically also see that many neopeptides arise from mutations in hotspots. So these are marked by these vertical lines here. So these mutations are very well known to be recurrent in the different cancer types. So again, if you have a lot of mutations, you can generate more peptides. And then again, we have here just the accumulation of the frequencies of the peptides we have. And then also again, the CBio portal annotation, where we can see these clear spikes across the hotspot mutations as well. To better understand how well the different TP53 mutations are studied, we compared the mutation frequency to how often peptides from those mutations were evaluated in actual studies. So here we have, so each dot is a mutation, and we can see here the number of PubMed IDs for each mutation, and then here we have the mutation and frequency in CBio portal. We can see there's a strong positive correlation that shows that more frequently mutated sites tend to be studied more often, as shown also before with these hotspot mutations. But however, we have here like a group of outliers, perhaps. So these are very common in tumors, so they are mutated in over 100 patients, but they have only been studied in one publication. So this points to potential research gaps and an opportunity for other research groups to maybe focus on these mutations and see they are recurrent in a lot of patients. What is the actual immune response for those? So next, we investigated the landscape of neopeptides and the fraction of validated neoepitopes across the different cancer types. As I mentioned, we had over 28 cancer types, but for this analysis, I pulled some of them together and I only looked for the organ side. For example, as I said, like breast cancer, for example, I only said it's breast cancer and didn't distinguish between the different subtypes of breast cancer, and then same for the other cancer types. So as I expected, cancers that are known to have a high mutational burden, like colorectal cancer, skin cancer, and lung cancer, they dominated the data set. They have the most peptides reported. But when we look at the percentage of peptides that were actually recognized by T cells, so the green parts of these bars, the picture is a bit more nuanced. So colorectal cancer has a relatively low neoepitope positivity rate, which is also plotted here. So it's just 6% of the epitopes were positive. But skin and lung cancer had significantly higher rates. So this makes also sense clinically because the skin and lung cancers, they also respond better to immunotherapies. So to account for the differences in mutational burden across these cancer types, we also normalized the neoepitope positivity fraction by the median number of mutations per cancer. So again, we looked at the CBioportal portal and we checked the mutational frequency they have, and then we used this number to normalize the mutational frequencies. So after normalization, we saw some important shifts. For example, colorectal cancers normalized positivity rate jumped to 39%. So this might suggest that its low raw positivity rate we saw earlier was more a reflection of a high mutation burden than a lack of T cell recognition. So that the positive epitopes, they get diluted by the high number of mutations. A similar pattern was seen in lung cancer, where again we have the jump, but we don't have that jump in skin cancer. Some interesting observations we can also see in low mutation burden cancers like bile duct and pancreatic. So they had a very low mutational, they have really low mutational burdens, and the positivity rate is also lower. But when we normalize this, we can see they have quite some significant higher positivity rates. And this is surprising because these are cold tumors, but it could mean that while you only have a few mutations in these cancers, when mutations actually occur, they may be more likely to be recognized by T cells. So in conclusion, we performed the largest meta-analysis to date of experimentally validated neoepitopes. We uncovered some patterns and biases, and there's also some research gaps. So these are opportunities for us researchers to dig into and do more experiments on. And this paper was recently published in Cancer Immunology and Immunotherapy. So you can go and look at this paper to get all the details about the analysis. And you can also download the data that was used. So we have the supplemental tables in a very clean way that you can also download and do your own analysis. So in this work, a lot of people were involved from LJI and also from the UCSD and in our collaborators in Denmark. So I want to thank all of them, and then also the funding NCI and the ITCR funding mechanism of NCI. And yeah, as before, please post your questions into the Q&A, and then we can talk about it. Thank you.
Wonderful. Thank you so much, Zeynep, for presenting that work. The prostate cancer meta-analysis, I think, was done last year, and the Neoepitope one this year. So it really is hot off the press. It's very exciting to be able to show that to everyone. And also demonstrate what you can do with the CEDAR database, the power of having all of the experimentally derived epitopes or the assays that they were tested in concentrated into one database with captured with a lot of granular detail. So you can conduct things like meta-analyses and look at where the research is and perhaps where the gaps are in the research and focus your attention there if this is your area of interest. But we are like running 4 minutes ahead of schedule, so we'll just add that to the Q&A because we are moving into section 2 Q&A. So I would like to invite Dan, Zeynep, and Bjorn back to the virtual stage to start our section 2 Q&A. And I can see, Dan, you've selected a question, so maybe I'll let you take the first one, unless
Yeah, I
Bjorn.
can take the first one. That's fine. So the question is, how does PepX look up the peptide in different databases? Each database has different identifiers, timestamp. How do you locate the peptides? So we actually took the ensemble GRCH38 reference genome and generated every possible peptide above eight, or sorry, including more than 8 amino acids. So it's actually just like a giant lookup table across these different databases, but the tool also combines the gene symbols and TPM values for within each data set as well. So that's essentially how it works. Right now it's fixed at that reference, but it may be updated eventually. Bjorn, do you want to take the next one?
Thanks, Dan. I'll take the next one. Anonymous attendee asks, you showed in the first day the growth in IEDB. I wonder you noticed the growth helping in known biases like HLA coverage. So yes, as the IEDB has grown over time, like HLA coverage is a good example. So like how many epitopes do we have that are reported to be restricted by different HLA molecule? With the continuous growth of data in the IEDB, we have seen that more and more HLA alleles are being covered. At the same time, the biases are still there. So there is still going to be more data on HLA-A0201 out there just because there's more reagents to study it, there's more tetramers for it or so, and people are going to be having these tools available to them to do their experiments that then kind of reinforce getting more data for this very well studied allele. So it's not that the growth necessarily means that we see less biases in data in terms of the relative amounts, but by having now a million or I don't know, we're at 7 million or something like assays now rather than say a few thousands at the start, the number of alleles where there's at least one assay reported has grown substantially. And also over time, I mean, tools have been improved, and what used to be previously a necessity to focus on one allele at a time is just not there anymore. So definitely, I think over time, the increased amount of data has made biases less impactful. Zeynep?
Yeah. Can a neopeptide include many epitopes? What's the length range of neopeptides? So yes, a neopeptide can have many epitopes, as we saw in the figure with the TP53. We had many overlapping epitopes, so usually one mutation can generate many different neoepitopes. And the length usually, yeah, I didn't show those details, so the data is very skewed against the 9-mer peptides because those are the main class 1 binding peptides. But we also have a very long neopeptides, for example, from our own studies. We focus on 20-mer peptides, so we can capture all the different nested peptides within the longer peptide. And there's like some specific groups, they use 30-mer peptides. So you can see if like some group brings up a lot of studies, they will have like, suddenly you get a lot of 30-mers, suddenly you get a lot of 20-mers. But most of the data are like shorter peptides.
I can answer this follow-up question
Mm.
about PEPX. So the question is, for the lookup table, you need to take care of cancer mutations. So you create in silico peptides to make all possible combinations. No, actually, we don't, because you would have so much data, it'd be impossible to store every possible mutation. So that's why when I showed the pipeline, we used the reference um, column for looking up the expression values. And also, I should say too that like cancer mutations are sometimes also very patient specific. So if you generate these new peptides from a VCF file that's specific to a tumor sample, then it will map them back to a reference peptide, and then those can be looked up in PepX as an idea of how expression would be seen. Um, but yeah, it would just be not feasible to generate every possible peptide.
Yeah, I guess like how Dan touched on it, you can first use PepX, and there you can, not PepX, sorry, PepMatch. You can input your mutated peptides into PepMatch. It will find the reference proteins, and then those reference proteins from PepMatch you can use as input for PepX. So that's the way we recommend.
Right.
We
And I
have
would also
plans
add
to
to,
like combine it, make a combined tool, but right now we can do it with the pipeline, and
well,
it will
and you
work
can
well.
actually do that in the pipeline. I didn't show that, but you
Yeah.
could look up in PepMatch using one mismatch or two mismatch, then get all those match sequences and then search that in PepX. So that
Yes.
would be a good workflow to use as well.
Yeah.
Yeah.
Yeah, we should make a help article in our solution center, Nina, for
Hmm.
that.
That'd be cool.
Okay, what questions? Okay, maybe I can take there's also follow-up questions. If a neopeptide includes many epitopes, then this neopeptide is called a neoepitope, and then this neoepitope can include many epitopes. Yeah, so we look at the specific, we look at the short peptide. So if the short peptide has been tested, we say this is the short peptide. So if it can be that the 20-mer was tested in some context and it was negative, but then some nested peptides within those longer peptides were tested and it was positive, then that will be annotated positive. So we don't go and look for the nested peptides within a longer one and then use those assays to classify the longer one. That's something that you can do with some downstream analysis when you download the complete data set.
So
Okay.
there's another follow-up on the PepX lookup table. Can we use it as a replacement for BLAST or PepMatch? What would be the advantage of still using PepMatch or BLAST? So I guess it's maybe a bit of a misunderstanding on how the tools like work together. So PepMatch is trying to find matches within a set of proteins or a proteome. So you have a peptide and you search exact match or more, including mismatches, and you can get all possible matches within a reference. So you can get from one peptide hundreds of different matches across the proteome, potentially. But what PepX is doing is it's just looking up the expression values amongst these different data sets and combining, let's say it's found in, like I showed earlier, there was one peptide with like almost 100 genes. It combines those TPM values together into one value because it's expressed that much. So it's just a matter of your inputs and outputs. So you're taking a peptide and trying to find matches versus taking a peptide and trying to find TPM expression values. Bjorn, do you want to take that one?
Yeah, I'll take the next question. CCC 4002 asks, how frequent are the neoepitopes due to somatic mutations derived from histones presented to class 1 in some cancers? And then a separate question, are these histone neoepitopes correlated with the epigenetic changes measured in some cancers? So the first question is, there is actually, I just Googled for it, I searched CEDAR for it, and found a lot of histone data in CEDAR. And so this is definitely something that happens, and we definitely do have neoepitopes in histones. Now, the second question is kind of like outside of the scope of what CEDAR would be cataloging. We do not catalog epigenetic changes, and I think this is a fascinating hypothesis that you might have changes in histones leading to changes in, but that would be quite global at the epigenetic level. That might be then leading to different behaviors of cancer cells. So it's definitely an interesting hypothesis. I think where CEDAR could help with analyzing something like this is to identify where are there histones that are mutated in cancer that are then recognized by immune responses. Now, if you generally want to look for histone mutations, probably it's better to look at other databases that catalog mutations in general without necessarily the immune response, because based on how the question is phrased, it seems like you don't necessarily need the recognition of neoepitopes from those histones, but it looks like what you're really interested in is like how mutations in histones might shape the epigenetic landscape. So, I mean, obviously, we always love people using CEDAR, but you have to use the right tool for the right job. If indeed your question is mainly about where are mutations located and does that correlate with epigenetic changes, probably you're better served by more general cancer mutation databases, something like Civic. And I think that's it for me.
Yeah, I think those are all the questions.
Excellent. Thank you all so much. Thank you, Bjorn, Zeynep, and Dan, for your presentations and answering those questions. We are out of questions, and I believe a few questions have been answered with a typed response as well, just to give a little bit of extra detail. So definitely check those out to see if your question was one of those. But otherwise, thank you again. And now we'll move into our break. It was to be about a 15-minute break, but we'll take a little bit longer, just given we're ahead of time by about 5 or 7 minutes. So yeah, we will stop there. And that really brings us to the end of the first resource that we wanted, this first specialized resource that we wanted to talk about today, so CEDAR. Because after the break, we will be discussing the Los Alamos National Lab resources, the HIV databases, and the CatNap tool. And as I mentioned in the introduction, I've obviously presented this as a HIV resource, but the team has mentioned that their tools can actually be used for many other organisms as well. So don't want to pigeonhole them too much. So definitely stick around to learn more about what LANL has to offer as well. So with that, we will take about a 20-minute break. And I'll pop the timing in the chat as well. We'll come back at 10:30 a.m. Pacific time to move in our last few presentations for the day. Thank you all and see you in about 20 minutes.